Now, there are two angles to the story of the origin of eukaryotes from their prokaryotic progenitors. Each side is really its own story, and each story has an interesting personality from the relatively recent history of biology behind it. The endosymbiosis narrative must forever be associated with Lynn Margulis, a figure whose fingerprints are already all over the previous lecture, when we pointed out that the great oxygenation was catastrophic for the forms of life that had existed beforehand. There's no way to know how massive was the extinction that this precipitated, compared with the more familiar mass extinctions that are documented by the fossils of the Phanerozoic. Margulis was also famously cynical about the future of humanity, but not the future of life on Earth. Her point was that it's really not too likely for any species to have any fate other than extinction with no descendant taxa, and it's unsubstantiated arrogance on the part of our species, humans, to think that we might be any different. Life, on the other hand, is extremely resilient, and it will continue, largely in the form of microbial life, until the Earth gets incinerated with the expansion of the Sun billions of years from now. Margulis was also the first to seriously push the idea that mitochondria and other eukaryotic organelles, including peroxisomes, plastids, and eukaryotic flagella, they derived from bacteria that were symbiotic with a larger host cell. Her arguments were largely dismissed as fringy by established science, until they were no longer dismissible thanks to the growing onslaught of evidence provided by Margulis and others. Lynn Margulis was really an inspiration to me, and I'm happy to have met her when she passed through the University of Chicago while I was there as a grad student. Christian de Duve is the other name worth knowing in this lecture. De Duve received a Nobel Prize for his work on the origin and organization of cells, and I credit him for that part of the narrative accounting for the other parts of eukaryotic cell structure not explained by endosymbiosis. This largely consists of what we usually refer to as the endomembrane system: nuclear envelope, endoplasmic reticula, Golgi, vesicles, vacuoles. These are intracellular membranes that physically isolate different compartments in the cytoplasm and allow for functionalities that would not be possible in a non-compartmentalized cytoplasm. De Duve is also kind of interesting because of the way that he died. Euthanasia in 2013. The fact that euthanasia is legal in Belgium was eye-opening to me when I heard the news of his death, and it seemed just like a highly dignified end-of-life option that's not available to us in the United States. We should start with de Duve's part of the story because this would likely have preceded endosymbiosis. The question is really, how do we get from a small cell without any internal complexity to a large cell possessing extensive intracellular membrane enclosed spaces? The logic is actually very simple, and it breaks down into steps. First, we need to talk briefly about a theme that's usually developed as a standard lesson in introductory biology classes: surface area to volume ratio, and why cells are small. Now, if you haven't heard this one, the general idea is that if a cell gets its nutrients from the extracellular space by diffusion across a cellular membrane, it's the surface area of the outside membrane, the one that's in contact with the outside world, that determines the rate of nutrient uptake. The volume of cytoplasm inside the cell represents the biomass that must be fed by its membrane, and therefore having a high ratio of cell membrane to cell volume, or a high surface area to volume ratio, is a good thing because the cell's contents remain very well fed, and thus it can more rapidly convert nutrients into biomass. Other things being equal, a smaller cell has a larger surface area to volume ratio compared with a larger cell, and therefore the smaller cell has an advantage when it comes to taking up nutrients and converting them into more cells. Now visualize a head-to-head competition between a smaller bacterium and a larger bacterium. You could put 1 gram of each into a vat of nutrients. The smaller cell might double in mass every 20 minutes, while the larger cell takes 30 minutes to double its mass. In 1 hour, there would be 8 grams of the smaller bacterium, but only 4 grams of the larger. In 2 hours, the gap would be larger, The smaller cell would be at 64 grams and the larger at 16. And the gap would continue to get larger and larger, and thus come back in a few days and you would see that the smaller cell would be winning hands down this competition. Now we use this argument in intro classes to explain why cells are small. Growing larger comes at a cost of reducing the surface area to volume ratio, and becoming too large is actually a really bad thing. At the same time, you could argue that larger cells have other advantages that might be realized under other conditions. Take, for example, the absence of nutrients. A larger cell will contain a larger quantity of resources that could be tapped through a period of starvation, making it more durable than a smaller cell. In this regard, it's good to be larger because you're better able to withstand periods of starvation. Dew's model starts by offering a way in which a cell might be able to take the advantages of larger size without giving up surface area to volume ratio. He argues that with a more sculpted cell surface, for example with lots of involutions, say, you could keep a high surface area even as you increase in size. Dew then walks us through the next step in his story. If you have a cell with deep involutions, those involutions would serve as traps for organic matter, much in the way that your belly button forms a trap for lint. And the organic matter trapped in these involutions could be digested with the secretion of enzymes into the involutions, and then the nutrients can be absorbed. Dew sees this as the step just before the origin of phagocytosis, which occurred when the bottom of the involution packed with organic debris got pinched off and formed a vacuole inside the cell. Now, instead of secreting enzymes into the bottom of an involution, the enzymes get secreted into this food vacuole, and this is basically the form of cellular eating seen in a great diversity of eukaryotic cells, including the phagocytes that we have as part of our own immune defenses. When it first appeared, phagocytosis would have been a brand new ecological niche for larger cells, feeding primarily in this way, as opposed to the ancestral mode of simply absorbing nutrients by diffusion. Okay, so that takes care of the existence of larger phagocytic cells that may have been around in the late Archean, early Proterozoic. And here's where the Margulis side of things kicks into gear. We've already talked about how the great oxidation made things tough for living cells. Presumably, this includes some of the larger phagocytic cells, like the ones we've been talking about so far. The new presence of oxygen would have been bad for such cells that did not have the protections like catalase. Now, at the same time, there would have been some aerobic cells, like the alpha proteobacterial mitochondrial progenitor, a bacterium that was not only protected against the O2, it actively took up O2 with respiratory pigments like cytochrome for its use in ATP metabolism. Now, what if such an aerobic bacterium somehow ended up inside of the anaerobic archaeal host cell, perhaps by way of phagocytosis, somehow avoiding being digested? Now, this could have been a situation that would have been great for both parties. The bacterium would have found itself inside of a cytoplasm rich in the anaerobic end products that could have been used as food. The archaeal host would have benefited by having a living sponge to remove the O2 as quickly as it diffused into its cytoplasm. This is the basic scenario that we envision as having given rise to what eventually became the very first eukaryotic cell and its mitochondrion. The plastids, which include all of the eukaryotic photosynthetic cells' chloroplasts, would have come about a bit later, but by a similar endosymbiosis. The eukaryotic host took in a cyanobacterial cell, which eventually became an integral part of the cell's organization as the chloroplast. Now, the story of the origin of the mitochondria in the chloroplast is easy enough for us to visualize, but this isn't enough for endosymbiosis to have become a canon in biology texts. The evidence is an equally important part of this narrative because it is what lets us know that this is the true history behind these organelles, that they were once free-living bacteria. The fact that mitochondria and plastids have their own genomes is a very compelling piece of this evidence. When you look at the genomes of these organelles, their organization is that of a bacterium, not a eukaryote. The chromosome is circular rather than linear, and the genes remaining in these highly reduced cytoplasmic genomes match closely with those found in proteobacteria or cyanobacteria, respectively. These organelles also have their own ribosomes that match in size and structure with the bacterial ribosome, not the eukaryotic ribosome, which is about twice the size of the ribosome found either in the bacteria or these organelles. Both mitochondria and chloroplasts undergo their own cycles of binary fission and growth. That's independent of the cell cycle, mitosis and interphase, of the eukaryotic cell that contains them. As a scientist, you have to be able to support your story when the evidence is available, and there's plenty of evidence when it comes to the origin of mitochondria and plastids. Other eukaryotic structures may also have derived from endosymbiotic bacteria, but without retaining the bits of evidence we have in the case of plastids or mitochondria. Peroxisomes, for example, are mitochondria-like compartments that contain powerful enzymes that destroy bits of cellular waste via oxidation, but without any production of ATP energy. It's nearly a certainty that peroxisomes have an endosymbiotic origin similar to that of mitochondria, but given that peroxisomes are packed with such destructive enzymatic force, it's clear that this would not be a great place to house DNA or ribosomes. Whatever genes that are necessary for the continued production of peroxisomes must have been relocated to the nuclear genome. Lynn Margulis was a strong proponent for the spirochete origins from both flagella and the cilia that you find in eukaryotic cells. The ultrastructure for both of these is about the same, and Margulis even coined a new term for them. She called them undulopodia, in part to eliminate the confusion between them and the true flagellum, the structure that we find in bacteria, which we discussed in a previous lecture. Eukaryotic flagella and cilia are much larger than true flagella, and they actually generate their own motion internally, and not by means of a cellular wheel at their base. They are also intracellular, being entirely covered by plasma membrane. Margulis saw this arrangement as having originated from spirochetes attaching at one end to a larger cell, and with them continuing to undulate, this resulted in the larger cell being pushed around. I'll get to how Margulis got this idea in the next lecture. One point here is that there is, over evolutionary time, some fluidity to where genes get housed in the organization of eukaryotic cell. Some of the genes required for mitochondrial function are in the mitochondrial genome, but others have been relocated into the cell nucleus. Consequently, the nuclear genome of eukaryotic cells is a historic record of past endosymbioses experienced in the history of the cell lineage. The genes from a past endosymbiont could continue to persist in the nucleus, even though the physical structure of the endosymbiont or organelle has been lost entirely. We actually use this to support the idea that every known eukaryote derives from a common ancestor that had mitochondria, even though there are some eukaryotes that lack mitochondria today. By the same approach, we can confirm that the genomic evidence for chloroplasts is not universal in the eukarya, and therefore we conclude that many eukaryotic lineages evolved entirely without any history of having plastids. And in fact, it's only one of the eukaryotic lineages, the Archaeplastida, that took in a cyanobacterial endosymbiont to become the prototypical photosynthetic eukaryote. We humans are part of one of the lineages that never had plastids.